we said, an exam already? That's really early. And as I hear that you have exams next week, hopefully you appreciate that you have one under your belt. And in order to talk about enzymes and enzyme mechanisms, we really have to make sure you know the amino acids and structure, because we're going to assume you know, if I say a serine side chain, you can vision it in your head. You know, is it going to be nucleophilic? Is it going to have an ionizable proton, etc.? And so that's why we kind of, okay, why do I see a different screen there? No. Okay, I see a slide that you don't. Okay. Okay, there we go. And when I looked, so I made like the YouTube recording of the last 2 lectures, and it's like, wow, that's really pathetic. 5 slides in an hour. And so, just a little bit. Okay, so I think I'm recording. It said it was continuing recording, right? Okay. Hopefully I really am. And I know I haven't uploaded the last few. You haven't cared anyway, but you'll start to care soon, I think. Okay, so we're going to talk about enzymes, and I think nobody's going to be surprised that enzymes are the biological catalysts, the majority of biological catalysts. So are proteins the only macromolecules that do chemistry? What other macromolecules can do chemistry? RNA. Okay, in what context can RNA do chemistry? Okay, there's RNA enzymes. So where did you learn that? Cell bio. Good job, cell bio. Who taught cell bio? Dr. Tagg or Dr. Muday? Okay. So until 20 years ago, nobody knew that RNA did catalysis, and the party line was, if chemistry is happening in the cell, a protein is doing it. So it turns out most of the time, if chemistry is happening in a cell, a protein is doing it. But there is this narrow subset of RNA molecules that do chemistry. We're not going to talk about them anymore. It's just to increase your vast knowledge of biomolecules. Okay, so enzymes, on the whole, are proteins. So they speed up reactions. We're going to talk about the mild conditions. So if you're in an organic chemistry lab, how might you make a reaction go? What might you do to the reaction? Heat it up. What else?
High pressure.
Add high pressure, change the pH pretty significantly, microwave it. Okay, all kinds of things you can do to a reaction that you can't do in a biological system. Okay, so basically enzymes, again, are boring organic chemistry. So we have a very narrow range of pH, temperature, pressure, and so enzymes actually do chemistry that's more sophisticated often than we can do in the organic chemistry lab under limited conditions. So that's kind of the contrast there. So if we think about how enzymes catalyze reactions, so this is kind of the generic enzymatic reaction. Okay, so an enzyme binds a substrate. So what's a substrate? Okay, so it's a ligand, so substrate is to enzyme as ligand is to protein, right? So the substrate is going to bind the protein. We'll talk about some of the complementarity between the substrate and the enzyme in a minute. But how is a substrate different from a ligand? Okay, it's going to bind in an active site. We could say that ligands bind in... Okay, how does it change? Change does chemistry to it, right? Okay, so if you look on the right side of the equilibrium, we don't see a substrate over here, we see a product. Okay, so chemistry happens to the substrate to turn it into a product. Ligands don't really change. It's just they bind, they release. They bind, they release. Okay, and so we're going to see that when an enzyme binds a substrate, the first thing it has to do, though, is bind to it, right? So that part of it is kind of like ligand binding. It's binding, but then here's chemistry. Okay, so if we say the first equilibrium is binding, then we have some chemistry, then we have release. And it's not quite as simple as binding. So when we talked about proteins and ligand, what was the K that we used to describe that interaction? KD. Okay, so we're going to see that often when we talk about enzyme reactions, we don't use KD because that doesn't include the chemistry part. So I'm kind of getting ahead of myself a little bit, but kind of think about that distinction between ligands and substrates. Okay, so before we talk about the details about what kinds of, more details about enzyme reactions, we're going to talk about classifying the enzymes. So scientists like to classify things, right? Kingdom, phylum, whatever, that not being a biologist I can never remember. I guess I learned it in 10th grade and then never went back to it. So you know that if a name of something ends with ASE, you can make a pretty good guess that it's an enzyme, right? But then biochemists, enzymologists, like to group things, okay, like all scientists. And so we're going to try to group every enzymatic reaction into one of 6 categories based on the kind of chemistry that happens. I don't care that you know what the numbers are, but I do care that you know what class of enzymes these belong to. So you need to understand the chemistry, but I don't care that you know that lyases are class 4. Okay? However, biochemists care that lyases are class 4 because when you classify an enzyme, you give it a 4-number name. So EC stands for Enzyme Commission. So just like in chemistry, we have the IUPAC, International Union of Pure and Applied Chemistry, and that names things systematically, right? So for enzymology, we have the Enzyme Commission. So if we were to discover a new enzymatic function, so we discovered an organism we never knew before, and all of a sudden there's an enzyme that we've never seen in any other organism, you could still probably classify it into one of these 6. If you find something outside these 6, that's kind of mind-blowing, but you better be able to classify it into one of these 6. So if it has hydrolase activity, the first number there would be a 3. So the next number in the Enzyme Commission number would be, a subclass, and then subclass, and then the actual, what substrate does it use in this class of enzyme. Okay, so every enzyme has it's not 4 digits, but it's a 4-number code that's kind of like a barcode for that enzyme function. So we're going to go through each of these. You'll see enzyme names. You don't need to match up these enzyme names, but these are all enzymes we're going to see throughout the rest of the semester. So from now on, every enzyme we talk about, whether it's in glycolysis or the citric acid cycle, we will talk about what class of enzyme is it. Okay, and so you don't have to learn these now because you'll learn them eventually. Okay, so the first class, oxidoreductases. So just from looking at the name, what do you think happens? Redox reactions. Okay, so there's going to be some electron transfer. So there's going to be something gets reduced, and then something else has to get oxidized. Okay, so down at the bottom are some of the hints as to names of enzymes that fall in this category. So a peroxidase, an oxygenase, a reductase. Those are all enzyme names that give you a hint. That's probably an oxidoreductase. Okay, so we're going to see lots of dehydrogenase enzymes. So you will know mechanisms for dehydrogenases before too long. And so how many of you have already worked with lactate dehydrogenase? Okay, so if you're taking the lab section of the class, you're working on lactate dehydrogenase, whether you know it or not, and you should know it. And so in this reaction, so the way you do it in biochemistry lab, you start with lactate as the substrate. You add the enzyme lactate dehydrogenase. So what's this NAD plus molecule? Okay, so we'll come back to that in a minute. It's a cofactor. Okay, and so it is going to get reduced. Lactate will get oxidized to pyruvate. So if you have an oxidoreductase, something gets oxidized, something else gets reduced to complete that redox pair. Okay, next class of enzyme are transferases. So some chemical functionality, some chemical moiety gets transferred from one substrate to another substrate. Okay, and so what is being transferred in this enzyme, alanine transferase? So the amine group. Okay, so this class of enzymes we would also call aminotransferases. Okay, so it's the interconversion between an amino acid, alpha ketoglutarate, and an alpha keto acid. Okay, so the alpha keto. So here is the. So there's the carboxylate. Here is the alpha keto group, and now once the aminotransferase happens, now glutamate is the amino acid, and pyruvate is the corresponding alpha keto acid. Okay, so it's an interconversion of an amino acid and alpha keto acid by moving the amine group around. Okay, and so you'll see other examples of these. So do you know a class of enzymes that is also a transferase where what gets transferred is a phosphate? Kinases. Okay, so another example of a transferase that you've already heard of, I think. So a kinase. Okay, so a kinase is an enzyme that takes a phosphate group, typically from ATP, and puts it onto something. Okay, so often kinase, and we'll see lots of examples of kinases too. So a kinase will often put a phosphate group onto something as a way of regulating enzyme activity. So when we talk about enzyme regulation, there are some enzymes that get their activity turned on by a phosphate group in a particular place, some that get their activity turned off by a phosphate group. So this class of kinases are very important in enzymatic regulation. Okay, so it's kind of like the control panel. Oh, we need a phosphate group on this enzyme, we're going to take it off of this enzyme, and so that will regulate lots of metabolic energy in an organism. So kinases are transferases. Third class are hydrolases. Okay, so a hydrolase, how would you define this? Oop. I guess I put it on there. So a hydrolase reaction is we're going to break a chemical bond, break a covalent bond, using a molecule of water. Okay? Can you think of any hydrolases that you already know the name of that you learned for the first exam? Say it again, Jim. Okay, so a condensation reaction would be the opposite. Okay, so a condensation reaction is you're pulling out molecule of water. Trypsin and chymotrypsin, very good. Okay, so trypsin and chymotrypsin break peptide bonds by adding a water molecule across that peptide bond. So again, that's the opposite of condensation, because when you make a peptide bond, it's a condensation, you basically pull a water molecule out. Okay, and if we go back to our friends, the kinases, so after a phosphate group is added by a kinase, you have to take it off, right? You don't want that enzyme always turned on, so if you're going to turn off the activity of a kinase, the opposite of that kinase activity is a phosphatase activity. So a phosphatase is also a hydrolase. Okay, this is a pyrophosphatase, so it's just a special example of a phosphatase. So this phosphatase is just cleaving that bond between the pyrophosphate group, so that's two inorganic phosphates that now separate. Okay, that's hydrolases. So the fourth class are called lyases. So this is the class that sometimes I have a hard time recognizing. So the so it's really making and breaking double bonds. So adding a group across a double bond, or removing a group to make a double bond. Okay, so often when we look at enzymes in class from now on, if we look at it and we're like, oh, I see a double bond going away, it must be a lyase. Okay, so lyases add groups across a double bond or remove groups to create a double bond. Okay, isomerases. This one's kind of easy because we all know what isomers are. So we're just rearranging parts of the same molecule. Okay, so one example is just interconversion of L and D amino acids. So L-alanine and D-alanine are isomers of each other. Stereoisomers, in fact. So the enzyme that interconverts them is an isomerase. You see plenty of those as well. Okay, and finally ligases. So ligases are my favorite class of enzyme because this is the class of enzyme we study in my lab. So if an enzyme has the name synthetase in it, so it means that bonds are made using a molecule of ATP. Okay, and this one might be a little bit hard to see, okay, because, so what happens to the ATP molecule that's on the left here? So it looks like it just has the phosphate group taken off. Okay, so it looks like, wow, what purpose does that serve? Okay, and so there are many enzymes that would not be energetically favorable to make their products in the absence of ATP energy. Okay, so remember, we all know that ATP is the energy currency of the cell. So do we just like light a match, like burn the ATP to get the energy? How do we capture that ATP energy to make this reaction happen, do you think? Okay, so we're going to break the bonds, and what's going to happen to those bonds?
You'll release energy.
Yeah, kind of. Okay, so ATP has high energy bonds, as we'll see later on. So I'll give you a clue. I'm going to draw the ATP, which you'll be able to do before too long, I promise. If you don't know how to do it already. With my big old Sharpie marker here. So this is the triphosphate of ATP. And here's our ribose. And take a shortcut and call that the adenine base. Okay, so the key for synthetases is usually they make an intermediate that uses part of the ATP molecule. So we're not just going to break that bond, we're actually going to attach part of the ATP to the substrate to make an intermediate. Okay, so again, you don't need to know the mechanism for this now, we'll see it later, but essentially that carboxylate side chain of the glutamate amino acid is going to attack the gamma phosphate. So in ATP we name these alpha, beta, gamma. So we're going to attack the gamma phosphate, open up that double bond, collapse it again, and break that bond. So now we have, and I know this is really messy, so this carboxylate side chain is now a phosphate ester. Okay, so that's the rest of the amino acid. And now we have that phosphate ester plus the ADP, right? And now what can the ammonium group do? So do you see how chemically we've essentially activated that carbonyl carbon? Okay, so now the amino group from the ammonium cation, so now it can come in and nucleophilically attack that carbonyl group, right? And you get opening of that double bond, collapse, and loss of the phosphate group. So the purpose of ATP, we're not just going to like break a bond and somehow the energy is used. So we're using that ATP bond energy to make an intermediate that activates that side chain for subsequent nucleophilic attack. Okay, so now we have the ADP left over, we cleave off the phosphate, and now the amine that was part of just the ammonia floating around is part of the amino acid. Does that kind of make sense? We'll come back to that, so if you kind of went, ooh, I'm not sure I got that, we'll definitely come back to it. I just want to point out that ATP is used in ligases to drive the reaction by making that covalent intermediate, which is a higher energy intermediate. Okay? Questions? Or are you willing to set that aside for now? Yes?
Doesn't it say condensation reaction, so where's the water molecule?
Yeah, so I guess condensation, when we say you remove a molecule of water, that's kind of, so we're condensing two things to make a new bond. It's not always water that comes out. So, sorry about that. Okay, so if those are 6 classes of enzymes, now we're going to come back to the substrate and the enzyme again, okay, on a more detailed level. So what is it that allows any given enzyme to bind its substrate? Okay, so before chemistry happens, the substrate has to bind. So kind of the same thing as for ligands. So something on the enzyme is going to be complementary to the substrate. So it could be charge, probably is shape, probably is size. And so the substrate is going to fit into a binding pocket. Okay, a binding pocket / the enzyme active site. They're not always the same thing. Okay, and so what do you think we mean by lock and key? So what's the lock and what's the key? The lock is the enzyme, the key is the substrate, it opens the lock, chemistry happens. Okay, and so how is that not always maybe a good way to describe it?
Changes confirmation.
Okay, so we're going to change, we might, when the enzyme binds the substrate, we might change conformation.
It's never a perfect fit.
Okay, why would you not want it to be a perfect fit?
This is never really too tight bonding.
Okay, so if an enzyme binds its substrate too tightly, what's going to be the driving force for chemistry to happen? Okay, so when the substrate binds to the enzyme and says, oh, I'm happy here, I fit so well, nothing is going to be the driving force for changing the substrate into product and that product's leaving. Okay, and so this is, that's why it's kind of in air quotes. We understand the concept of shape and geometric complementarity, but we don't want it to fit too tightly. Okay. In addition to shape and charge complementarity, we also have stereospecificity. Okay, so enzymes are specific for one enantiomer of a substrate. So when we talk about all the enzymes in glycolysis, those are specific for one class of sugars. Conveniently, it's the class of sugars that we have around us. And so we can't digest, so what are the naturally occurring sugars? D sugars. Yeah, so if the sugars that we consume are D sugars, we could consume an L sugar, but it's not going to do anything for us. So it's going to go right through. We're not going to get any nutritional benefit because the enzymes aren't set up to to react with those substrates. Okay, and then the last kind of add-on to the enzymes, often proteins and enzymes have extra things that help make them work. And so I tend to use the words cofactors and coenzymes interchangeably. I'm sure there's a better definition. But so they might be metal ions, they might be things like a heme group, they might be cofactors like NAD that help the enzymatic reaction, but aren't part of the enzyme. So proteaceous, I don't know, what's the word? Non-protein parts of the enzyme reaction. So they're not substrates and they're not proteins. So what are some of those? So certainly heme groups, flavins, all kinds of things. So coenzymes are organic molecules, but again, sometimes I call NAD a cofactor and sometimes I call it an enzyme. It's really a coenzyme. I don't care if you call an ion, if you call the NAD a cofactor, we kind of use that in a slightly sloppy way. It's really a coenzyme. Okay, so, but part of the key is if anything happens to that coenzyme, it has to be regenerated for the full catalytic cycle to be in effect. So just like any catalyst. So here's our NAD plus. Yes, Andrew.
So if it's a metal associated with an organic complex, is that under coenzyme?
So I would say, and here's where the semantics get a little bit fuzzy. So for example, if you have a heme group that's part of an enzyme, obviously you're going to have the metal there. So is the whole heme plus iron the coenzyme? Yes. Do you have other enzymes that have an iron that's not part of a prosthetic group or part of a cofactor, and then it's just part of a coenzyme and then it's a cofactor? That's why some of us use the terms kind of loosely. So I would not make a big deal about that. So certainly any non-organic additive, like a metal ion, would not be a coenzyme, it would be a cofactor. So you wouldn't call just a zinc that's part of a catalytic site in an enzyme, you wouldn't call it a coenzyme. Yeah, you call it a metal cofactor. Yeah, so it's the kind of thing, unfortunately, that's not a very good definition. It's just as people start talking, you kind of realize, oh, that's how you use the word. So kind of like if you've read a word and you've never heard it pronounced, and then someone says it, you're like, oh, that's how you say that word. Yeah, so same thing with cofactors and coenzymes. We'll start to use them, and then you're like, oh, she always refers to NADH as a cofactor, but I know it's really organic, so it's probably a coenzyme. Yeah, it's not a critical big deal. Okay, so it is important what they do, though. So NAD plus, NADH, again, I would probably use the word cofactor, but really they're coenzymes. Nicotinamide adenine dinucleotide. So because there are two dinucleotide bits, do we ever draw the whole thing? No. We draw what we call the business end of the molecule. Okay, so when we start doing dehydrogenase reactions, we never draw the bottom part, simply because it would take up all the paper, and we're really concerned about the part where hydride groups add. So, again, we'll come back to the function of some of those dehydrogenases, but, for example, NAD plus, NADH is involved in hydride transfer reactions. Okay, and so what's a hydride? So it's an H minus, which always freaks us all out, right? Because we're used to protons, or H plus, right? So a hydride, merely just a hydrogen atom with an extra electron. So this is the kind of chemistry that if you do it in an organic chemistry lab, you really have to beat on the molecule to get hydride transfer to happen, right? So sodium borohydride, all kinds of crazy stuff. But the enzyme says, sure, I'll take a hydride off that molecule and put it on that one. I don't care. So the hydride gets added to that position, and then we rearrange, right? Okay, so NAD plus, NADH, here's the hydride that we added. And again, we'll do lots of those reactions later on. Okay, so many of the building blocks of coenzymes and cofactors that we need for metabolic reactions are in our breakfast cereal. Okay, so often the vitamins, so what's the definition of a vitamin? necessarily. Some of them are inorganic. So if we skip that part. So what are vitamins? So a vitamin has kind of a circular functional definition. So a vitamin is any compound for which if it's missing in your diet, there's some defect. So some functional defect for the organism that is reversed by adding it back into your diet. Okay, so essentially vitamins are essential nutrients for which if you don't have them in your diet, there's some implication. So some phenotype, essentially. Chris? That's only from micronutrients. So like a protein is not a vitamin. Yes. What would it be if it wasn't a micronutrient? No, just tell me why you ask. I'm not used to thinking in terms of micronutrients. So a macronutrient would be like sugars and proteins and fats. Okay, yeah. Okay, so we should clarify. A micronutrient that you need in your diet. Yeah, so I guess essential amino acids wouldn't be vitamins. Yeah. From a nutrition standpoint. Okay. But if you look at, so usually it's the ones below the lines, right, are the micronutrients, I guess. Maybe I need to start using that word. I like that. So, for example, NAD. So what's the component of NAD that's the vitamin? So niacin. Okay, so niacin is the building block for nicotinamide in NAD. We'll see plenty of other cofactors as we go through that use the others. So thiamine, so there's an enzyme cofactor called TPP, thiamine pyrophosphate, and it's often used in carboxylation, decarboxylation reactions in metabolism. Look, there's thiamine. So vitamin A, vitamin C are often involved in chemical reactions, and so that's why they're classified as vitamins. Yep. Folic acid, we'll see folate as a cofactor, etc. So we need to consume them in our diet, often to make these coenzymes that we need for metabolic reactions. Okay, so what is it that enzymes do functionally? And so if they promote a reaction, okay, so if this is our uncatalyzed reaction, the substrate is going to go on to be a product. Okay, so we have some free energy of the ground state, in this case some lower free energy of the products, okay, and we have to, so what do we have to overcome in the middle? So some activation energy barrier, right? So we could heat this up, so we have to do something to get the reaction to go. And so what does the enzyme do? So it lowers the activation energy barrier. So just like any other catalyst, right? So it does not change the substrate and product free energy level. Okay, it just lowers, so it's like you're chopping off the mountain in between, right? Don't tell my PChem colleagues I said that. We're not chopping off the mountain. We're lowering the activation energy barrier. Okay, so now we have some additional part of the reaction, which is the enzyme substrate complex. Okay, and you see, we talked earlier about the enzyme substrate complex, where the enzyme and substrate have to find each other. Then chemistry happens. Then you have the enzyme product complex. We often kind of use that shorthand of enzyme substrate and kind of ignore that enzyme product complex. That's often very short-lived. We often can't capture it, and so we know it's there, but we don't really talk about it. Okay, so how well do enzymes work? So, yes.
Previous slide. Which step is rate limited?
So it depends. It depends on the enzyme. Okay, so some enzymes are for some enzymes, product release is actually rate limiting. For some, the chemistry in between is rate limiting. Okay, and so it really depends. Which of these transition states is higher. Yep, and it totally depends. For some, even just interaction with the substrate could be rate limiting. Often binding is not rate limiting. It's usually chemistry or product release. Good question. Okay, so how well do enzymes work? And so some of these, I don't even know how they come up with these numbers. So how would you measure a reaction that's 10 to the 17 slower than an enzymatic reaction? I think you're just kind of extrapolating out. So some enzymes speed up reactions like catastrophically fast, dramatically. Okay, and then once we think about the fact that they do catalysis, how do they do it? And so these are the 5 main classes of ways that enzymes catalyze reactions. So sometimes I call these catalytic strategies. Okay, and we're going to say that the first 3, so enzymes might use one or more of these. So some enzymes will use general acid-base catalysis and some won't. A lot do. Not that many use metal ion catalysis, maybe only a third of enzymes. Not every enzyme uses covalent catalysis. We'll talk about what these mean in a bit. But, so that's some enzymes use those. And we're going to say that all enzymes do these last 3, and we're going to explain those. Question? Okay, so the first one, general acid-base catalysis. Okay, so just as we said, if you're going to make a reaction happen in the lab, you might change the pH. Okay, so often that will facilitate a reaction. If we can't change the pH in the protein, we might need the protein to act as an acid or a base. Okay, so if we just look at the top, and so if this is hydrolysis, so you can fit that on there, of an amide. Okay, so hopefully you can see at the top there we have an amide bond, so maybe it's a peptide bond, and now we're going to have an alcohol group come in and hydrolyze that amide bond. So again, what reaction might that you think of for that? An enzyme that you already know. So what enzymes do you know the names for already from exam 1? Trypsin and chymotrypsin? Yeah. Okay, so trypsin and chymotrypsin we're going to see. So maybe you didn't know the left half of the reaction, but you certainly know the right half, right? So it's cleaving a peptide bond, so it's breaking an amide bond. Okay, so you didn't know yet how that happened. You will. But I will tell you it's an alcohol group breaking a peptide bond. Okay, so if we're going to break that peptide bond, is this generation of charges in the middle there, is that a favorable situation? No. Okay, so we don't really want to generate a positive charge on an oxygen. That's not so cool. Okay, so what would make that reaction happen faster? Well, so what if we could add an acid or a base, right? So what if we could deprotonate that red proton there? Would that make the reaction go faster? It probably would, okay? Because now we don't have that positively charged oxygen. That's like the kiss of death for a reaction. Okay, so we could speed up that reaction in the presence of an acid or a base. Again, an enzyme is not going to do that. So we don't want a whole lot of acid or base in our enzyme. But what if we use this? So this is the beginning of enzyme kind of nomenclature. So this B double dot represents a general base. So if a base is an OH minus, for example, what would a general base be? Yeah, so sodium hydroxide would be a base, would generate a base. So in an enzyme, so if a base is going to remove a proton, so like a hydroxide ion can remove a proton, so a general base, in enzymology we would say that's anything present in the reaction that can remove a proton, but it may not be a hydroxide group. So knowing what you know about amino acids, what part of the protein could remove a proton, do you think? Okay, so a deprotonated lysine or arginine could, or a deprotonated aspartic acid, glutamic acid maybe, or a deprotonated, what would be the best amino acid to use for this? So which amino acid is very easily protonated and deprotonated in a neutral situation? Histidine, very good. Okay, so a histidine side chain could be a very good, you should always think the first general acid, general base would be histidine. Okay, so when we talk about enzyme reactions, a general acid is anything that can donate a proton. A general base is anything that can abstract a proton. Okay, so any of these side chains could be a general acid if it starts protonated, or it could be a general base if it starts deprotonated. But histidine is the one that's kind of happiest in either situation. Does that make sense? And we took kind of a long way to get there. Okay, so if in the active site of an enzyme you have a deprotonated aspartic acid, so how readily is that going to take up a proton? So what pKa for aspartic acid, for example? So 3.6 is a pKa. Is that likely to take up a proton at neutral pH? Not so likely, but we can mess with the pKa's in enzymes. So even though you learn the pKa, turns out in enzymes they fluctuate. So depending on the need of the enzyme, you might have an aspartic acid that becomes protonated even though you don't really predict it from the pKa. So the pKa of 3.6 that you learned, maybe the pKa of that one glutamic acid or aspartic acid is more like 6.5. Okay, so the pKa's can shift very widely, and it's not every aspartic acid in the protein, it's just the one in the active site. Okay? So even though we've learned the pKa's, they can shift pretty dramatically. Okay, so that's acid-base catalysis. So again, we're going to see lots of acid-base catalysis. Most of the enzymes that we see are going to use acid-base catalysis in some form. So the next catalytic strategy is covalent catalysis. And covalent catalysis is when in the course of the reaction, so during the reaction coordinate, there's a new covalent bond made between the enzyme and some intermediate. Okay, so there's covalent bond made. It can't stay like that because, of course, the enzyme has to come back to its starting conditions at the end of the reaction. But for some time period, there's a covalent bond between the enzyme and the substrate or intermediate. Okay, and so here now you're going to see the chemistry of chymotrypsin. So you know what chymotrypsin does, but now you're going to see how it does what it does. Okay, so this kind of goes along with the other slide. So we have a serine residue in the active site of chymotrypsin. So what's the pK of serine?
14.
Over 14. Okay? You're thinking cysteine. Good there. Because there are actually cysteine proteases. So chymotrypsin is a serine protease. We're going to spend a lot of time learning about serine proteases. But they're named serine proteases because they use a serine residue to affect catalysis. There's also cysteine proteases. So, but this serine residue gets deprotonated. So what does that mean about its pKa? It's dramatically downshifted. Okay? So we have a general base. So chymotrypsin uses general acid-base catalysis and covalent catalysis. So I will tell you that, so what would you expect is the general base in chymotrypsin? Histidine, very good, it actually is. I don't know why they just don't write it in there. So histidine is the general base that takes off the proton of serine, the serine side chain, to make serine. So what job is the serine side chain doing in that reaction? So it's the nucleophile. Okay, so this is nucleophilic attack. Okay, so deprotonation, as we saw in general acid-base catalysis, deprotonation makes that serine a better nucleophile. Okay, so now the serine side chain acts as a nucleophile. Okay, ultimately what happens, so this is kind of a lot of shorthand, but it opens up, then it closes. We're going to see this later in excruciating detail, I promise you. And then we break the peptide bond, and what are we left with, at least for a little while? So you have an ester. So the intermediate part of the substrate is attached to that serine side chain, so this is covalent catalysis. Okay, so chymotrypsin, and also I'll tell you trypsin is the same. Trypsin and chymotrypsin use covalent catalysis. So the active site serine side chain becomes covalently incorporated into that intermediate. Okay? So we're going to see lots of enzymes that use covalent catalysis. Metal ion catalysis, again, so it's not the majority of enzymes, only about a third of them. So the ion could be there to promote a water molecule to be more reactive. It could be positioning a substrate. It could be a structural metal. So again, it could be being oxidized or reduced. So in the electron transport chain, we see lots of metal enzymes, because the metal can be a redox center. Okay, and then the last two catalytic strategies are used essentially by all enzymes. Okay, so the first one is called proximity and orientation. So what does proximity mean? Just nearness, okay? And orientation, just making things line up right. Okay, and so this is kind of, when you, once I explain it, hopefully you'll say, well, of course that's what all enzymes do. So all enzymes bring the substrate to get, to itself. Maybe there's two substrates that are part of the reaction, and then we bring both substrates together and position them in the right way for chemistry to happen. Okay, so chemistry can't happen on the enzyme active site if the substrate isn't there. Okay, so proximity is just you're bringing the substrate there. It's going to be near that serine side chain, for example. So proximity. And then orientation. So if that bond to be broken is not in the right orientation, it's going to be hard for chemistry to happen. So inline attack of the nucleophile, for example. Okay, so all enzymes have to bind the substrate and then position it properly for chemistry to happen. And so here's just a little example. So if we have an ester and a carboxylate coming together to make an aldehyde, so how fast will that happen? Well, it will depend on the concentration of the two molecules, right? It will depend on the temperature, maybe. So you got to bring those things together for chemistry to happen. And not only do they have to come together, they have to come together in the right orientation. So if we now connect the ester and the carboxylate, so that now it's a unimolecular reaction. Okay, so a first order rate constant. Now that's going to be sped up. So it's going to be sped up 5 orders of magnitude. Okay, so what's that, 100,000 times. Simply because you're not bringing two things together, they're already there, they just have to rotate and find the right orientation. And now if they're connected and kind of forced into the right orientation, now that speeds it up another 1,000-fold. Okay, so that's what enzymes do. They provide a platform for the substrate to come in, bind in the right orientation for chemistry to happen. And when we looked before about how well do enzymes work, so kind of 10 to the 8th rate enhancement is a pretty average number for enzymes. So really a lot of the rate enhancement comes from the simplistic idea of just bringing things together. Okay, and then transition state stabilization. That's the last catalytic strategy that, again, I would say all enzymes use. And so this gets back to the lock and key model and how it's not a great description for enzyme catalysis. So this is kind of a silly example of the catalysis we want to happen is breaking a metal stick. Okay, so we have to bend it and break it, and now we have a broken metal stick, metal rod, whatever. And so in the absence of an enzyme, you have to put in some energy to get over the transition state, and then you have products. Okay, so the product energy is lower than the substrate energy, but there's a big energetic barrier in between. And we could say, oh, lock and key model, let's have an enzyme that fits the substrate. Okay, so if it's a metal stick, you maybe want an enzyme that has magnets surrounding a binding site. Okay, now the enzyme will certainly bind the substrate tightly, but what's going to happen? Nothing. The enzyme's going to bind the substrate tightly, right? And there's going to be no chemistry. So now we've actually made an enzyme substrate complex that's too stable. Okay, so we've stabilized that enzyme substrate complex, and now we've actually increased the energetic barrier. So we've made it even harder for chemistry to happen. Okay, so now it just kind of can't randomly flex. It's locked into that straight position, and it's never going to break. Okay, but now if we have the enzyme, instead of looking like or fitting the substrate, now what if we had an enzyme that fit the transition state? So if the transition state is this bent metal rod, now what if the enzyme active site has magnets lining a cavity that forces kind of the metal stick to be bent? Okay, so now the enzyme looks like the transition state instead of looking like the substrate. Okay, and so now there's a driving force to bend the stick, get over that transition state barrier, over that energetic barrier, and now we didn't change the substrate or product. We have a not overly stable enzyme substrate complex. We've lowered the activation energy barrier. Does that make sense? Okay, so enzyme active sites often look like their transition states. So enzymes will bind transition states in order to drive the reaction forward so chemistry can happen. Okay, and there's 2 bits of evidence for this. Okay, and I always get to this point when everybody's kind of glazed over, and then they kind of look at me like, huh? What are you saying? So let's see if we can, so sit tight and see if this will help you understand transition state stabilization. Okay, so what if you want to make a drug? Okay, so we have drug discovery class. You're like, okay, I want to make a drug. Not Breaking Bad kind of drugs, but like, and you want to make an enzyme inhibitor drug, okay? And so do you want to make a molecule that looks like the substrate and binds instead of the substrate? That might be one way to start, but it would be better if you could synthesize a molecule that looks like the transition state. Okay, so if you can make a molecule to look like the predicted, because we can't always solve the structures of transition states, if you can predict what the structure of the transition state of the reaction is, then synthesize a molecule that looks like it. Now what's the enzyme going to bind? Is it going to bind the transition state analog or the substrate? It's probably going to bind the artificial transition state analog that you made in the lab and not bind the substrate, and therefore it's not going to do any chemistry to the substrate. And so now that transition state analog is a good enzyme inhibitor and then hopefully a good drug. Does that make sense? So a lot of drug discovery programs are really chemistry predicting what that transition state is, and then how would I make a molecule that has that geometry, that shape, that charge, but no chemistry will happen to it. So it's going to bind in the enzyme active site and no chemistry will happen, and therefore we've turned off that enzyme activity. So that's the first bit of evidence. The second one is cool to understand once you understand it, so let's see if we can get there. So if we come back to the same idea of transition state analogs. So a synthetic molecule that looks like what you've predicted the transition state of the reaction to look like. Okay, so you make a molecule in the lab based on what you've predicted on paper. Now you have that transition state analog, that organic compound, and you inject it into a rabbit, for example. What's the rabbit going to do with that? Make antibodies, right. So the rabbit's immune system says, that's a foreign object, I need to get rid of it, so I'm going to make antibodies. So what do antibodies look like? So we always draw them as Ys. So if you're in the chemistry seminar on Wednesday, the poor guy's like, I don't know why they draw them as Ys. I'm like, because they look like Ys. But so they look like Ys, right? And so every antibody has two binding sites for their antigen, or what they're supposed to recognize, okay? And so the antigen, we're going to draw it as a little molecule there. So it will bind to the antibody, and that's a very tight interaction. Okay, so now we have this transition state analog. We think that's the energetic barrier for chemistry happening. We inject that into a rabbit. The rabbit makes antibodies. We purify the antibodies out of the rabbit's blood. And now what do we have? We have an antibody that binds a transition state analog. Instead of now mixing in the test tube the transition state analog with the antibody, what if now we have the antibody and we add a substrate to it? What will the antibody do to that substrate? So it's going to bind this substrate, but is it going to look like the substrate when it binds? No, the antibody is going to kind of pull it along the reaction coordinate so that now the substrate looks more like the transition state because that's what the antibody wants to bind. Okay, so it's going to pull the substrate to look like the transition state, and then what happens after it's the transition state? Then it goes downhill to products, right? So we've kind of gotten over that energetic barrier to make, substrate goes on to make product in the presence of an antibody. Okay, so now what did the antibody just become? A catalyst, an enzyme. So kind of an artificial enzyme. Okay, and so this is called catalytic antibodies. So basically artificial or engineered enzymes. Okay, and so catalytic antibodies are not as good as natural enzymes because all they do is binding. There's no acid-base catalysis. There's no, it's not that there couldn't be, but that's not necessarily happening. It's really just you're selected for just binding, not for chemistry. So it covers one of those categories, but only one. Okay, so it binds the transition state, but may or may not do covalent catalysis or acid-base catalysis. But the fact that it works at all is pretty impressive. Does that make sense? Do we do that slowly enough that, yeah, it's kind of an abstract concept, but if you can kind of picture the little bunny with things stuck into it and then pull out the antibodies and do some chemistry. Okay, we have a minute maybe for questions. Does anyone have questions? Otherwise, I'm happy to stick around for a few minutes. And so we've made it through kind of the introductory enzyme stuff, and now we'll talk about mechanisms on Monday. So we're going to spend a lot of time with chymotrypsin. So chymotrypsin will be your friend.
So it's all, yeah.